Trish, another drug was recently approved for GIST as well, and that would be avapritinib. Could you discuss a little bit about its mechanism of action and the evidence that supported its FDA approval?
Absolutely. So avapritinib and ripretinib are the two drugs that have gone through some relatively parallel development. They have some commonalities in terms of how active they were in the unique subset of the PDGFRA D842V mutant subset, whereas we described earlier, imatinib doesn't work, and there's really very marginal to limited activity of any of the other kinase inhibitors that we have been describing. So the true unmet need was real, for example, in that patient population who had absolutely no standard of care treatment. So avapritinib tested in a single arm cohort of 50 plus patients had a pretty phenomenal response rate that was upwards of 70%, disease control rate that was upwards of 80 to 90%, and a median survival that was not reached at the time when the analysis was presented and the FDA looked at the drug and the application. So I think because of the phenomenal activity in that particular subset, despite the single arm nature, no randomization, I think as the FDA would like to say, clinical benefit was meaningful enough that the drug got its indication in specifically that D842V mutant subset for the PDGFR alpha group. There's also been evidence of activity of the drug in the KIT mutant subsets. I think it is selectively active in the more common secondary mutations in the KIT mutant subset, exon 17, exon 18, for example. It's important to note that even that, as homogeneous as we think that population may be, that's still a heterogeneous population given the exact sites of mutation in the exon 17 and 18 and the changes in the amino acid sequences, if you will. So in the third, fourth line setting, in the KIT mutant subset, avapritinib had a response rate of 22%, which is actually very impressive. That's a high RECIST response rate. Patients who responded did have a durable response because the median duration of response was upwards of 10 months. I think the catch is that the overall median PFS, as we have been discussing, turned out to be 3.8 months. And I think my interpretation of that data is that the drug is very active in a specific mutant subset. So where it works great, and there is durable benefit, but there is enough of the other population where it has marginal or limited activity, which sort of dragged down the median PFS, if you will. And this is sort of the phase 2 data that was telling us these numbers. And probably an extrapolation, or that explains why the Voyager trial, the randomized phase 3 study comparing avapritinib to regorafenib in the third line setting, there was a press release recently, and it's been public knowledge now, that the primary endpoint of progression-free survival improvement was not reached and is not likely to be reached, and therefore the trial has been halted. And along those same lines, efforts at trying to move the drug in earlier line settings in the second line also likewise have been halted. But coming back a full circle on a positive note, I think avapritinib, from an efficacy standpoint, certainly has defined activity and therefore approval in the D842V PDGFRA subset. And it does have defined activity in certain specific mutations in exon 17 and 18 that will need to be defined a little bit better for it to be of any use. From a side effect and toxicity standpoint, I think some of the common GI toxicities were relatively well managed or manageable. I think one of the unique toxicities of avapritinib was this cognitive defects. There were some neurologic toxicities, including some intracranial hemorrhages and memory losses that were an area of concern. I think there was an active investigation in terms of what's causing it, how can we predict it to try to allow for better management of those things. And I think that still will be relevant in the D842V population where the drug has dramatic activity and is a commercially available agent.
Yeah, I think that was a great summary. I would add a few comments. One is the cognitive toxicity that we're seeing, I feel like we're seeing less of now than we did initially. And I think that that's probably because of greater recognition of this as a potential side effect, earlier detection, and then either dose modification or interruption or some other strategy to improve it. So I do think for people who are prescribing it for the D842V mutant tumors, there needs to be really careful attention to cognitive changes and early management of that. I think that makes a big difference in the severity of that toxicity. The other two other comments. One, I totally agree that there is activity of avapritinib in KIT mutant GIST. A response rate of, I think, 17% in the Voyager study is really impressive. But the work that needs to be done is to figure out who those patients are or what tumors are responding. Difficult to do from archival tissue, and hopefully we can make some use out of circulating tumor DNA or other approaches to understand which patients were responding to treatment and to see if there's still a strategy for using this medication to help control disease or even generate responses. I mean, 17% is higher than we saw, certainly sunitinib and regorafenib, and even higher than what was reported with ripretinib.
And I think it's fair to comment that while we have paid more attention to PFSs all along, in this later line settings, response rates are not trivial. I mean, I think patients with bulky disease who see some significant volumetric reduction of the tumor, it does allow for better symptom management. It absolutely impacts on quality of life and their ability to carry on a normal day-to-day routine. So it is absolutely not trivial. And I agree with your additional comments.
Yeah, and the median PFS is a point, is one spot among 100% of patients. That's the 50th percentile. So, you know, if there's a tail that has a good response, and if it's durable, then that is an area of activity. The other comment I'd like to make is just that the benefit that was seen in the D842V population is noteworthy. And I would make the analogy to imatinib for KIT, you know, standard KIT mutant GIST. Both of these situations was a disease that had no effective treatment. So before imatinib, there was no treatment for GIST, for metastatic GIST. Before avapritinib, there was no treatment for D842V mutant GIST. And so I think that is something that should be celebrated, which is that there is, for a rare patient population, that there has been a drug developed and that it is effective and it can be safely administered and managed, and it is life-changing for this patient population. So I do think that is something worthy of celebration.
Absolutely. A non-medical term that's appropriate here is that they're both game changers in that population of patients, right? It's no question. Yes.